Hi, my name is Cassie Argenbright, and under the guidance of my mentor, Dr. Perry Fuchs, I recently had the opportunity to do a study investigating the relationship between hyperbaric oxygen therapy and anxiodepressive pain behaviors in an animal model of fibromyalgia. For those who don't know, fibromyalgia is a chronic widespread pain disorder that's generally of a non-inflammatory nature, and it's characterized by this lack of tissue damage. And it's really considered an affective disorder due to its super high prevalence alongside depression, anxiety, sleep disturbances, cognitive deficits, and many more comorbidities. More than 2% of the US suffers from fibromyalgia, and on average, patients spend about $1,500 a year looking for alternative treatment modalities outside of the typical clinical pain management regimen. This could be for many reasons. As you can see in figure 1, we really identify pain to be this multidimensional experience that includes not just sensory components, but the affective or emotional components of pain and cognitive components as well. Therefore, effective pain treatments should treat on a sensory level as well as affective and cognitive. Animal models really provide us a unique and vital opportunity to investigate pain disorders like fibromyalgia. And so we wanted to investigate how well an animal model of fibromyalgia pain replicated the pain experience on both a sensory and an affective level. There's also been a lot of growing evidence in clinical populations for hyperbaric oxygen therapy as a potential fibromyalgia treatment, both sensory and emotionally. So this study served as the first study investigating the role of hyperbaric for fibromyalgia in the United States in both preclinical and clinical literature. So we utilized 48 female Sprague Dawley rats and randomized them to a fibromyalgia condition or a control condition, and a hyperbaric oxygen therapy treatment condition or a controlled treatment condition. So we utilized the acidic saline model developed by Sluka et al. In 2001 to induce the fibromyalgia-like pain by giving two 4.0 pH saline injections into the left gastrocnemius muscle 5 days apart to develop a bilateral hyperalgesia. As you can see in figure 2, the pre-treatment column right here shows a reduced threshold in both the blue bar and the orange bar, which signifies animals in the fibromyalgia conditions showing greater pain behavior than controls before treatment. So we can see the model worked in inducing this sort of fibromyalgia-like pain. Then animals in the hyperbaric oxygen therapy treatment condition were given 2 treatments of 100% oxygen at 2.0 atmospheric absolutes 24 hours apart, as signified by the blue bar in figure 2. And we expected this to raise the thresholds over time, signifying the alleviation of this fibromyalgia-like pain. But we did not see that here over time as we were expecting to in that blue bar. In terms of affective measures, we measured anhedonia or depressive-like behaviors by using a sucrose preference test, which exposed the animals to a 2% sucrose solution and a normal water solution in their home cages for 72 hours to see how their preference habits changed. We expected the animals in the fibromyalgia condition who did not get the hyperbaric oxygen therapy treatment. In figure 3, these are these orange bars across time. We expected those animals to show the least sucrose preference, indicating more depressive-like behaviors. However, as we can see over time here, there are no between group differences despite pain or treatment condition, which is not what we had expected to see. Lastly, to measure anxiety-like behaviors, we used an open field test where animals are placed in a chamber and their locomotion is measured for 5 minutes to determine how much time they spend in either the center of the apparatus versus along the perimeter. Animals who spend more time along the perimeter of the apparatus are showing more anxiety-like behavior. We expected the animals in the fibromyalgia condition who did not get hyperbaric oxygen therapy, again, this orange bar in this graph right here, to show the most anxiety-like behavior by spending the most time around the outside on this right side of the graph and the least amount of time inside, which is right here on this left side of the graph. But again, we didn't see at all what we'd expected to see here. There were no between group differences in time spent in the center or the outside of the chamber for any conditions. But again, like I said, we did not see what we'd expected to. So overall, this study showed that we have seen continued validity for this animal model of fibromyalgia in inducing that sensory pain associated with fibromyalgia-like pain, as we can see from figure 2. But it did not produce the affective comorbidities that are required to be diagnosed with fibromyalgia as a disorder. Additionally, we didn't see that hyperbaric oxygen therapy alleviated the fibromyalgia-like pain like we'd expected it to. Both of these findings really contradict the current literature, and we'd really like to direct future studies towards one, finding those experimental circumstances that the animal model does replicate the full fibromyalgia experience on both a sensory and affective level, and two, investigate the exposure response curve within this hyperbaric oxygen therapy treatment to identify what a more effective dosage might be for alleviating this fibromyalgia-like pain. Thank you.